Beneath the soil surface lies a very beautiful relationship that exists in nature, that is the mycorrhizal association. Hello everyone, my name is Sneha, PhD scholar from GV Pant University. Today we are going to discuss on the topic mycorrhizal association types and mechanisms of their benefit. Throughout this lecture, we will be briefly discussing about what is mycorrhiza, its different types. I mean, it really have several types and their mechanisms as well as their benefits. This topic is not only important for the microbiology perspective but also for the point of view of soil sustainability as well as soil management. Now, let's begin. Actually, the mycorrhiza is actually derived from the Greek word mycos, meaning fungus, and rhiza, meaning roots. So, this is actually a symbiotic association between the fungus as well as the root. So, this literally means fungus root, and it in plural way, mycorrhiza is actually termed as mycorrhizae. It was actually discovered by Frank in the year of 1855 in forest trees. Actually, he has observed the mycorrhizal association in the trees such as pine, and it was a very revolutionary thing at that time. Imagine the situation in 1855. At that point, everything was revolutionized because uh, earlier on they think that fungus were actually pathogen, but Frank told them the fungus is actually beneficial. So this was a challenging, or you can say controversial. So Frank pointed out that these are actually a symbiotic association. Actually, he point out that there is a symbiotic association with fungi that exists under the natural condition in the root system and other crop plant. So mycorrhiza is not only in the pine tree but also everywhere, and this was challenging. So why there is a symbiotic relationship? So there is a symbiotic relationship because imagine there is a plant and there is a fungus. Now, how there is a symbiotic relationship? Because the plant being green, so it can perform photosynthesis process. Now, as it perform photosynthesis, so now they can actually provide soluble carbon sources to the fungus, because they can perform photosynthesis, so they will provide carbon to the fungus. Now, as the fungus which cannot perform photosynthesis, so they actually are heavily dependent on the host plant. So in return, actually the fungus provide the host with increased capacity to absorb the water as well as nutrition. Now, how the imagine there is a tree and there is mycorrhizae, which is actually fungus because it literally means fungus root. Now the fungus root actually, it has hyphae, and hyphae is thinner as well as longer than the plant root. So because it is more thin and longer, so it can penetrate inside the soil pores, and it can go beyond the depletion zone and can absorb all the nutrition which are present. So in this way, the fungus can help the host plant, or they are both have a symbiotic association. There are basically, we all know that there are basically two kinds of mycorrhizae. One is ecto and one is endo. Now, ecto, we all know ecto, what does it mean? It means outside. So this actually is the outer mycorrhizae. So the ectomycorrhizae is the fungus which we will discuss it one by one, but right now there is just a brief description. So ectomycorrhizae is the fungus which complete encloses each feeder rootlet. So this can't be shown properly. Okay, now here ectomycorrhizae is actually where the fungus is actually in the outer surface of the root. and in endo it is actually lives inside, or there is an intracellular establishment between the root cell and the surrounding. So we will discuss this further. Right now you just all remember that ecto means outside, which penetrates outside or between the cells of the root cortex, and endo means inside. Now there are several types of mycorrhizae according to Smith and Reed. So if we see the types of mycorrhizae according to Smith and Reed, which has given in the year of 1997, there are seven kinds of mycorrhizae. So these all are the seven kinds, in which you can see the fungal taxa as well as plant taxa, but the intracellular colonization is only absent in ectomycorrhizae, and otherwise it is present. Why so? Because it is ecto, that means it has outside, so there is no colonization inside the cell. The fungal sheet. And what is fungal sheet? Fungal sheet is the outer outer surface of the hyphae. So it is actually present everywhere but not in arbuscular, and it is absent in arbuscular, and in ecto and arbitoid it is present or absent. And vesicles is only present in arbuscular. You all have heard about VAM, that is vesicular arbuscular mycorrhizae, and somewhere it is denoted as AM or somewhere it is denoted as VAM. So when it has vesicles, then it is known as VAM, and if it does not have vesicles, then it is called as AM. So it is present and absent, and in the rest of the things it is absent. Now the vesicles are absent. These all are different types according to Smith and Reed. This is the whole chart which actually denotes the type, the distribution of different kinds of mycorrhizae in globally. So the major contribution is AM, which is arbuscular mycorrhizae, which accounts for 72%. I mean, this is a lot, and we will discuss it why this accounts a larger global scale, because there must be a reason, right? Like human population is highest, there is a reason that we are intelligent, or there are other factor, like there is survival of the fittest. But we will discuss about why arbuscular mycorrhizae is highest. Now the next one is orchids, which accounts for 10%, and the rest you can see. Now there is non-mycorrhizae and AM, NM. Non-mycorrhizae 8%, and AM, NM is 7%. Now AM, NM is arbuscular mycorrhizae and non-mycorrhizae. Now what is the difference between AM and this AM and NM and this NM? Actually, here these are those kinds of mycorrhizae that exist in nature, where these are normally non-mycorrhizae, but under special condition. Under special condition, they can convert themselves into arbuscular mycorrhizae. So in normal condition, it is non-mycorrhizae, and if there are certain several conditions, then it will transform itself into arbuscular mycorrhizae. So these account for 7%, and the rest you can see here in the pie chart. Now we will discuss plants' dependence on mycorrhizae. So how the plant actually depend on mycorrhizae. So in nature there are actually various plant which actually depend on mycorrhizae. So earlier, now as there are many dependencies, so earlier scientists have given several terms for classification. So early researcher actually used the term mycotrophy, which was given by Rommel in the year of 1939, which means actually which tells actually the meaning of nutrition of plants through the fungi. However, this term are actually general, because it only tells that how much the nutrition is actually provided to the plant. It actually tells that the nutrition has been transferred to the plant via fungi, but it actually does not tell us how much the degree of dependency. I mean, how much how much degree in which the plant is dependent on the fungi. or the soil fertility which is influenced, or the variation among the plant species. So for more clarity as well as for precision, new terms were introduced. So the first one is mycorrhizal dependency, which was termed by German in the year of 1975, which defined as the degree to which the plant. So here they does not explain the degree of dependency, but in mycorrhizal dependency it defined the degree to which, I mean how much, the degree to which the plant depends on the mycorrhizal association to achieve maximum growth at a given level of the soil fertility. So how much degree the plant actually depend on the fungi. The next one is mycorrhizal responsiveness, which was given by Mossy in the year of 1973, which represent the growth response of the plant after the mycorrhizal colonization. So the term itself define itself. So it tells how the plant is actually respond after the after the mycorrhiza colonization. So several other terms were also described. The first one is relative field mycorrhizal dependency, which actually tells the dependency of mycorrhizae under the field condition. The mycorrhizal efficiency ratio, which tells how efficiently the mycorrhizal improves the growth, because we will discuss like the mycorrhizae improves the growth. The mycorrhizae inoculation effects. Now the effect of the plant after the inoculation of mycorrhizae. Next slide. The ecological significance. So we will see here the significance of mycorrhizae. So mycorrhizae actually varies with soil fertility being higher in low phosphorus soil. So first have a two condition where there is a low phosphorus condition, and there is a one condition where there is high phosphorus condition, and in both the cases we have mycorrhizae in that. But where there is high phosphorus content, now the plant will actually the plant actually does not need mycorrhizae to extract all the phosphorus, rather as it has its own root, so they will extract the phosphorus which is present in the soil pool. But when we have low phosphorus content, then the plant actually depend more on mycorrhizae. So you can understand both the difference. Imagine imagine you have suppose I'm not getting the exact example now. Suppose you have lots of money. Now you have your own money. Okay, you have your own money, a lot of money, then you will depend on your parents lesser. But if you don't have money enough uh, during your school time, so you will depend more on your parents. So in similar way, the plant is the parent and the child is the mycorrhizae. So if it has lots of money, that means it has lots of phosphorus content, then they will extract lots of phosphorus, then the dependency will be less. But if it has less amount of phosphorus, now the plant actually need more amount of mycorrhizae, or they are more dependent on mycorrhizae. So the availability of the mic, and this is the first point, I hope you all understood. Now, the availability of mycorrhizae propagules such as spores, hyphae, infected root fragments determine the plant establishment and competitions. So, if the propagules are absent or low, the mycorrhizal dependent plant fail to establish. That means that if the mycorrhizae does not mycorrhizae propagules, such as if the spores does not establish in the plant, or the hyphae does not establish, then how they will then the mycorrhizae will not grow. If the mycorrhizae will not grow, then the dependency will stop. So this is a easier point, uh, that actually in simpler term, If there will be no mycorrhizae, then how come there will be dependency? If there will be mycorrhizae, then only there will be dependency, in very simple terms. Now, mycorrhizal dependent plants compete poorly in nutrient deficient soil in absence of fungi. So the mycorrhizae dependent plant. Now, if a plant is dependent in mycorrhizae, it actually compete poorly in nutrition deficient soil in the absence of fungi. That means if a plant is dependent on sorry if a plant is actually dependent on the mycorrhizae, that means it cannot extract the nutrient without the mycorrhizae. And if it does not have a proper nutrition around them, that means suppose if there is a plant and it is dependent on mycorrhizae, that means it has poor root, or the root length is shorter. Now it depends on mycorrhizae to extract all those nutrients. So now if the mycorrhizae is absent and we have a nutrition deficient condition, now the plant will die, because how come they will extract all the nutrition which is present inside the soil? They cannot. So this the point is actually explaining this point only, that the mycorrhizal dependent plant compete poorly, that means they cannot compete easily in the nutrient deficient soil in the absence of fungi. If there is no fungi, how come they compete each other? Low specificity. Actually, low specificity means that, here what low specificity means, if there is one fungi and there is one fungi and it can infect several plants, and vice versa. That means if there is one plant, there are several types of fungi that we have seen, ecto, am, or orchids. There are several other fungi. So there are several other types of fungi and there is one plant, so it has low specificity. If there will be high specificity, that means high specificity, that means one plant and there will be only fungi. So this is the condition. So there is low specificity, that means one fungi and several plant, and vice versa, one plant and several fungi that exist between the plant and the mycorrhizal fungi, which promotes diversity. So you understand how there is diversity: one plant several fungi, and one fungi, or one fungi several plant. So in this way there is diversity, stability, because when there is diversity there is stability. And with an example, like if there is suppose if there is only monoculture, if we are only practicing monoculture in agriculture, then will be less stability as compared to if we have diverse system of crop. If we'll have different type of cropping system, then it has more stability as compared to the monoculture practice. And evolutionary success of the plant communities. Now there will be evolution of the plant communities. There are different kinds of plant community. The mycorrhizal independent advanced plant with well-developed root system and abundant root hairs can compete effectively with mycorrhizal dependent species for growth and nutrition. That means if there are two plants and when there is one plant which is actually dependent on plant, and there is one plant which is which is not dependent on the mycorrhizae, they will both compete with each other. How? If the mycorrhizae independent plant has an advanced system of root. Now like imagine this is a root and these all are hyphae. Now let's say soil this is the soil portion where there is only nutrition, like in this space only there is nutrition. That means imagine only phosphorus is there. Now the root hairs, this is only the root hair, only this portion is the root hair, and the nutrition has been depleted. That means the root hairs has extracted all the nutrition. All right, now this is the this is the hyphae. Now the hyphae actually can go beyond the phosphorus depletion zone. This is the depletion zone. The hyphae penetrate outside the depletion zone. The root hair cannot penetrate outside it, but the hyphae can penetrate, which explain that uh, because if a plant is heavily dependent on the hyphae, if they are heavily dependent on hyphae, that means they actually they actually can survive in phosphorus deficient condition. But suppose uh, if this is hyphae matlab, this is a mycorrhizae independent plant, they does not depend on mycorrhizae. So this condition can only be valid if the root hairs will be strong. If we have longer root hairs, we have thicker root hairs which can extract all the nutrition which are present beyond the depletion zone. I hope you understand by this diagram, and if you don't understand, please go through this diagram, which is very important. Understood about how, now you all must have understood about how the mycorrhizal independent plant can actually compete efficiently with the mycorrhizal dependent plant. Now there is a key ecological feature of mycorrhiza is that there is actually the interspecies or intraspecies nutrition is being transferred from plants connected by the extramatrical hyphae. Now let us study one by one. So there is a intra as well as interspecies. That means within uh, within the same species the nutrition is being transferred. Suppose there is legume crop, then from one legume crop to another legume crop the nutrition is being transferred. But in intraspecies species transfer, the one is legume and one is cereal. Now the nutrition is being transferred from cereal to legume. I'm sorry, from legume to cereal. So in this way there is intraspecies transfer of nutrition, and this is only possible by the extramatrical hyphae. Now extramatrical hyphae is actually the external portion of hyphae which you have studied from the diagram. The nutri if Now this is the example. The nutrition is being transferred commonly which occurred between the coarse rooted legumes and the fine rooted grasses. The nutrition has been transferred from both of them. Now if the nutrition has been transferred and both the species are in long distance, all right, that means the distance is is longer, then the nutrition has been transferred and the carbon has been transferred from through the through the mycorrhizal network, and it is mainly done by the mass flow driven by the sourcing gradient. You all know mass flow and sourcing gradient, where there is a transfer of when there is transferred from higher supply to the higher demanding areas. So if there is a longer distance, then the nutrition is transferred via these process, and in mass flow there is actually there is total gradient transfer. And while if the distance is very smaller, then the nutrition is been transferred by diffusion and active transport. So if there is longer distance, then mass flow and sourcing gradient, and if the distance is less, then there is diffusion and active transport. Now, the plant response to the mycorrhizal inoculation is influenced more by the plant functional group and nitrogen fertilization. Like we have discussed about low phosphorus case. Now, if there is high nitrogen fertilization, so suppose if a plant has high nitrogen content. Suppose like this is the example, like non-N fixing forms. Woody plant and C3 plant respond positively than N fixing plant and C4 grasses. This is because in the first case actually they have lesser amount of nitrogen or a lesser amount of nutrition. So in this way they can actually they can actually respond more to mycorrhizae. Because imagine, like, there is a plant, and uh, if they does not, if it is actually, it does not, it is non-fixing, it has non-fixing, non-end fixing force. Suppose it has grasses, so it cannot do, it cannot fix the atmospheric nitrogen, so they depend on mycorrhizae. In the similar way, for C3 plant and for woody plants. Now, the physiology of mycorrhizae dependency. Now, the physiological dependency is mainly due to the improved phosphorus nutrition through the increased root surface area. We have discussed, like, mainly mycorrhiza is actually known for the transfer of phosphorus. Now in previous slide, like, there was for nitrogen. Now uh, later on we will discuss about the rhizobium. So actually rhizobium and mycorrhiza are actually interrelated. That's why the nitrogen portion was there in the previous slide. The mycorrhizas can transfer everything, but it mainly improve the phosphorus. So it, as it increase the root surface area, and because of the higher root surface area, they can extract more amount of phosphorus. If the surface area increased, the phosphorus uptake depend on the adsorptive surface area and reduction in diffusion distance of the phosphate ion in the soil. Now, why phosphorus is so important? Because actually phosphorus is immobile, and um, and uh, and the plant also depend more on phosphorus. So the mycorrhizae actually make it better, and the phosphorus uptake depend on the adsorptive surface area. And because the mycorrhizae increase the adsorptive surface area, adsorptive means how much it absorbs, and the surface area is also increased, and there is reduction in the diffusion distance of the phosphate ion. Because the distance is also reduced, the distance is reduced, the root length has increased, the external hyphae extend beyond the phosphorus depletion zone. We have studied about the depletion zone, like, there was a diagram. So the hyphae can extend beyond the phosphorus depletion zone, which we have studied. If now the mycorrhizae enhances the phosphorus uptake by exploiting the root rhizome not reached by the plant root. It again explained that the mycorrhizae actually enhances the plant root. The key predicts of mycorrhizae dependency. Now there are many several traits which is actually which actually include the mycorrhizal dependency. First is root and shoot length. Uh, suppose the the shoot is shorter but the root is longer. So do you think that the plant need mycorrhizae more? I have I'm going to explain it. Like, if the root length is longer, then why do they need mycorrhizae? Because as the root length is longer, they can easily extract the nutrition which is present inside the soil. But if the root length is shorter, if we are comparing it with shoot, then they need mycorrhizae, because as the root length is shorter, the mycorrhizae by its hyphae can actually extract the nutrition. So the dependency depend on the root and shoot ratio, the root hair length and the density. If the root hair length is longer, then they need less of mycorrhizae. And the density, I mean the root is thicker or thinner, etc. The phosphorus inflow rate, that means how much the phosphorus is been absorbed by the root length. If it can absorb more phosphorus, then it depend less on the mycorrhizae. But if it can absorb lesser amount, then they depend highly on mycorrhizae. Now the economic aspect. Now if the plant can extract the phosphorus easily, then we need not we don't have the need to apply more fertilizer. Now the fertilizer cost has been reduced, so this is more beneficial. So the economic dependency refers to the ability of the mycorrhizae to supply the phosphorus. Because it supply the phosphorus, so there is reduction in the fertilizer. All right. We also have to see that the extent of the phosphorus fertilizer substitution by the mycorrhizae without the yield loss represents the measure of the dependency. That means we also have to look upon the aspect that if we are applying the mycorrhizae in such a way that the yield must not be lost. Because what is our first motive? Our first motive is to improve, to make better things for the farmers. So if the yield is been reduced, so what the farmer will gain? So we have to see in the yield aspect, in the economic aspect, we have to make farmers wealthy. So in that way we have to substitute mycorrhiza. Suppose the mycorrhiza can extract uh, suppose if the plant need 60 kg of P2O5, all right, and the mycorrhizae can actually provide 30 kg of P2O5. Now we only need 50% of phosphorus fertilizer, because earlier we have to give more, now we have to give only shorter amount. So we cannot be heavily dependent on the mycorrhizae. In this condition the mycorrhizae is only providing 50%, so we need more fertilizer. Okay, if you say no, we will not do fertilizer, we have mycorrhizae, no, we also have to see the economic aspect also. So this last line is explaining that point only. Now the mycorrhizosphere. The rhizosphere, we all know the rhizosphere is actually the zone which is near the root zone, which is the root zone. So the zone near the root is called as rhizosphere. Now when there is root, there is mycorrhizae. If there is mycorrhizae, so if there is mycorrhizae, now the roots plus mycorrhizae is mycorrhizosphere. You understood? Like the zone around the root is rhizosphere, and root plus mycorrhizae is actually mycorrhizosphere. So the development of mycorrhizae, this is only the definition, that the development of mycorrhizae inside and outside the root leads to the formation of specialized zone. So inside and outside, because the mycorrhizae which can penetrate inside, what we call it, we call is endomycorrhizae. So the mycorrhizae which can penetrate inside the root is called as endomycorrhizae. So inside and outside both the when we calculate both of them, when we see both the aspects, so the development of mycorrhizae inside and outside leads to the formation of specialized zone, which is actually called as mycorrhizosphere. Now, the mycorrhizosphere actually influence the rhizosphere microflora by altering the host plant physiology and root exudation. Now, how they can influence the uh, root exudation pattern? Now, what is root exudation? These all are the chemicals which are being secreted by the root. Now, because of the mycorrhizosphere, there will be less amount of nutrition which will be needed by the root. There will be less stress as they will extract all the nutrition. So there will be less stress exudates which will be excreted by the roots. So they actually alter the pattern of the root exudates and the plant physiology also. The host plant genotypes plays a major role in determining the AM symbiosis. Now, what is the type of the host plant is also very important in determining the symbiosis. That how the symbiosis is being done by the here there is AM Arbuscular mycorrhizae. The edaphic means soil factor. The edaphic, environmental, cultural, and chemical factor actually influences the host physiology. Host physiology, that means the crop physiology. Now the edaphic means soil factor, that are the pH, EC, its porosity, texture, everything. Now they will influence the host, the plant, environment factor like climate, rainfall, humidity, etc. Cultural like tillage operation, cropping system, chemical factors like the fertilization, etc. It influences the host physiology and mycorrhizosphere interaction. So how the the mycorrhizosphere interaction has been done with the host, that means the plant. The root exudates supply carbon to the rhizosphere microorganisms. About 20% of the photosynthesis are released by the root exudates. Now this is an important point, that 20% of the photosynthesis, that means how much the plant actually perform photosynthesis. Now if there is, suppose if there are 100 carbon, 100 carbon which are being generated by the photosynthesis, 20 carbons are being excreted by the root exudates. All right. Now, the mycorrhizal colonization alters both the quality and the quantity of the root exudates that we have discussed, influencing the microbial population structure. Mycorrhizal actually influences both the quality as well as the quantity. That means how much the exudates will be released. So this will influence it. The redistribution of carbon through the extramatrical fungal hyphae affects both zymogenous and autochthonous microflora. Now, what is zymogenous? Zymogenous is actually those kind of microorganisms which actually feeds on the readily available carbon. So, if uh, the hyphae because the hyphae actually influence the root exudates, and now the because of the root exudates, because it is now easily available, so the zymogenous feed on them. They are actually rapid, but the autochthonous are not rapid, they are constant. So they are more dependent on the native soil microflora. So they depend on the native one. They are the constant, so the carbon which are being present inside the the soil pore space, they are very intact. Now the autochthonous microflora actually extract those. So the carbon which are being redistributed by the hyphae affects both of them. The mycorrhiza actually enhances the population of beneficial organisms, because the mycorrhizae increases more amount of nutrition. It increases the nitrogen fixers, the phosphorus solubilizing bacteria. It improves the plant growth and health. Mycorrhizal plant actually maintains the higher population of introduced beneficial microbes, because of more amount of nutrition which has been present again by the hyphae. So more amount of microbes will also be present. Mechanisms of colonization, which is very important, that which has every steps of how the am fungi actually colonization. So we have three stages. First is pre-symbiotic stages, that means there is no contact stage. In this stage we have three stages. The first one is pre-symbiotic stage, the second stage will be contact stage, um, will be here, I guess, here. And the third one will be establishment of am fungi. So the first one is the pre-symbiotic stage, that is the contact stage. So here, under suitable condition, the spore of am fungi actually germinate. So here the spores now starts to grow. So now there will be elongation of the fungal hyphae. The hyphae will also increase. First, there will be germination of the spores. Second, there will be elongation of the fungal hyphae. The hyphae will increase. Third, the hyphae will respire. They will start respiring. Now they are becoming live. Now the hyphae becoming live. Now they will call. Now they will call the plant. Now the calling system is actually known as the branching factor. So here the branching factor is actually uh, the branching factor is actually secreted by the plant root. They send signal. The plant root actually send the signal to the fungal ki okay, okay, please come. So this be my guest. So the plant root actually has the branching factor, and the chemically branching factor is actually the strygnolactans. These are the group of sesquiterpene lactones. So these are the chemicals. All right, so these are the signaling factors, like you have studied about quorum sensor. So similarly, these are the factors. So now in response, now the AM fungi actually calls back, and the response to them is actually MYC factor. So now they are responding. Okay, okay, okay. I'll be your guest now. So in that response, they actually release diffusible signal compounds, which are called as MYC factor. All right, now the contact has been established. Both have parties has called each other. Now they are both settled. All right, now the fungal hyphae actually reaches the root epidermis. They reaches the root epidermis through the contact recognition, like they have established, of the cell wall of the host hypodia, are formed from the extra radical hyphae. Now the hyphae through contact recognition actually form extra radical hyphae. Now after the formation of hypodium, like the host hypodium, the plant cell actively accept the hyphidial hyphae. Now because of the formation of hypodia, they now start accepting the extra radical hyphae, the hyphae which is present in the fungi. The nucleus of the epidermal cell attached to the hypodium migrates close to the hypodium and then moves to the opposite end of the cell. Now the nucleus of the epidermal cell, which is attached to the hypodium, actually migrates close to the hypodium. Like they get attached to the hypodium and now they move away from the cell, and tracing the nuclear movement exactly, the membrane of the ER, cytoplasmic microtubules and microfilaments are deposited and arranged to form tunnel-like structure. Now there will be a tunnel. That means the hyphae should move in such a way that they will not damage the plant root. The cells of the plant, like the hyphae will establish in such a way. Now the tunnel is known as PPA, which is the pre-penetration apparatus. A similar event will occur in the second plant cell layer before the plant root of the fungal hyphae reaches it. All right, it guide by guiding the hyphae through the region of weakened cell wall. Now they will guide through the region of weakened cell wall between the two plant cell, then the fungus. Now there is formation of arbuscular mycorrhizae within the cortical cell. The arbuscular now get surrounded by the plant plasma membrane derived the periarbuscular membrane. Now the arbuscular is actually surrounded by the plant plasma membrane derived from the periarbuscular membrane. Now the trunks of the arbuscular mycorrhizae actually undergoes repeated dichotomous branching. Now the trunk of arbuscular actually have repeated dichotomous. That means they have branching. All right, this is the case where they have branching and they will form mature arbuscular AM fungi. Now the growing and the maturation process of AM actually takes place only about 2 to 3 days, but the whole cycle from growing to death is actually for 9 to 10 days only. Now we have covered the mechanism. In the next, we will discuss about ectomycorrhizae, endomycorrhizae, and their different types. Now, if you have any doubt, please uh, feel free to comment, and you can give your doubt as well as your any suggestion. Thank you.